Hello, my name is Katerina Faya, and I will be presenting with Alexander Young about our article, which investigates the direct observation of the rotation of F1-ATPase. The diagram above demonstrated the three types of motors in cells, which are linear, rotation, and revolution motors, and they're demonstrated above. The motor which we will be discussing today, which is the rotation motor, moves around itself similar to a bicycle wheel rotating around its axis. Bionanimotors help make thermodynamically unfavorable processes possible. These include DNA replication, ATP synthesis, and muscle contraction, as they convert their energy supply into a rotary torque. So previous to this experiment, the only rotary motor that had been investigated was the one of bacterial flagellum, which is what allows bacteria to move. The motor moves clockwise to make the bacteria move forward and counterclockwise for a change in direction. The rotary motor of ATP synthase catalyzes the formation of ATP from ADP and phosphate, and when reversed, the hydrolysis of ATP. The F0 and F1 subunits of the ATPase shown in the image are both individual rotary motors, and the F1 subunit's function in ATP synthase is in accordance with the gamma subunit, and that's what's further analyzed in this paper. So this paper focuses on the F1-ATPase as previously mentioned, and this spans a total of 10 nanometers. The researchers' aim was to directly observe the rotary motor in the synthesis of ATP by attaching a fluorescent actin filament to the gamma subunit of the F1-ATP synthase in the respiring and photosynthetic cells, because this is the cells that it's located in the membranes. The hypothesis of the article was that the gamma subunit in F1-ATP synthase functions as a rotary motor central to the alpha-beta hexamer in ATP synthesis. One of the primary techniques used in this experiment was a process which anchored the subcomplex on a glass plate in order to observe the movement of an actin filament attached to the gamma subunit. This diagram represents the model used to fix the alpha-beta gamma subcomplex to the glass plate, and that's depicted by protein A. The diagram shows a PEG coating on the plate, but the experiment used a coating of horseradish peroxidase conjugated nickel NTA. The N-terminus of the 3-beta subunit of ATPase was linked with 10 histidine tags, which has a high affinity to the peroxidase, and that made the F1 side of the subunit bind more tightly to the plate. The F0 side of the subunit, otherwise known as the membrane side, is free from attachment to the glass, so it lets a fluorescently labeled actin filament be attached to the gamma subunit so its rotational behavior could be observed. Binding of the alpha-beta gamma subunit was dependent on the infusion of biotinylated subcomplexes and streptavidin. The rotation of the fluorescently labeled actin filaments attached to the gamma subunit on the F0 side was observed under an epifluorescence microscope. Epifluorescence microscopy is a method of fluorescence microscopy in which an excitation wavelength of light 546 nanometers, in this case, is passed on to the sample by passing through a dichroic filter cube that contains a fluorescence bandpass excitation filter. This allows specific wavelengths of light to pass through and reach the fluorescently labeled actin filament in the sample. This light is then reflected off of the dichroic mirror and through the objective lens. Once the light reaches through the sample, the fluorescent labeled fluoresces. The emitted light, which was measured to be in the range of 560 to 620 nanometers, passes back through the objective lens and is refined by the emission filter. The fluorescence of the actin filament is observed through the ocular piece of the microscope.
As shown in Figure A, the researchers placed an actin filament on top of the gamma subunit and observed anti-clockwise rotation when viewed from the membrane side. The researchers also recorded irregular back and forth motion in a minority of observed filaments rather than the counterclockwise rotation that the researchers expected. They attributed these random fluctuations to Brownian motion. These Brownian motions are observed in small particles suspended in the fluid and are theorized to be the result of molecules, in this case, impacting the observed filament. Figure B depicts the crystal structure of the F1-ATPase, also viewed from the membrane side. In this image, it is important to note the order of the different beta subunit forms and how the gamma subunit interacts with them. In this case, it's going from the empty form to the ADP form to the AMP-PMP, which is an ATP alternative form. This is analogous to the order of ATP hydrolysis, which takes place from the ATP form to the ADP form to the empty form. So direct observation of the motion of the F1-ATPase was completed using inverted microscope. This means that the observation was done from the bottom, formed a mirror image so that the image was shown through as though it was viewed from the top. The above images show various iterations of the data the researchers recorded, with image A showing the rotational process with the edge of the filament as the axis of rotation. Image B shows the same data. However, in this case, the axis of rotation is at the center on the gamma subunit. And image C is a trace of the centroid of the filament, with the bolded dark line representing the first revolution made. Researchers also noted filaments with lower average speeds and frequent pauses and fluctuations. These were attributed to obstructions by nearby proteins. However, they also mentioned that unresearched intrinsic rotary motor properties might be at play, which could also act as a basis for further research. The above data was adjusted to record revolutions per time through the researchers knowing the amount of time having passed between each image. The observed filaments were classified into 3 lengths: 1 micrometers, 2 micrometers, and 3 micrometers. And as shown above, the 1 micrometer filaments showed the highest rate of rotation. It's important to note on the right side of the chart how both the presence of ATP and the presence of a Z, which acts as an inhibitor of ATPase activity, both halted rotary motion. You can see this as the slopes of the lines on the right side are 0 for both negative ATP and plus a Z. So to conclude, the researchers definitively observed that the gamma subunit in the center of the alpha-3 beta-3 hexamer functions as the rotational shafts responsible for the driving of ATP synthesis. They also discovered the unidirectional rotation of the gamma subunit, which is an anti-clockwise rotation when viewed from the membrane side. So for potential avenues of further research could include investigating how can ATP concentration and the architecture of the overall motor affect the rotational speed and the generated torque of the gamma subunit. Furthermore, as discussed earlier, what sort of intrinsic properties of the F-ATPase rotational motor are at play with regards to regulating the rotational motion of the gamma subunit? So that concludes our presentation, and I'd like to open the floor to any questions.